Hey everyone, welcome back to my channel. I'm Princey and here we make a clinical research easy and exciting to learn. Let me ask you something. Did you know that every single medicine you take today, even a simple painkiller, was once tested in a clinical trial? Surprising, right? So, in this video, we are going on a journey through time. We all discover how clinical trials began hundreds of years ago, the mistakes that changed the medical history, and how we now use AI and digital tools to make a clinical trial faster and safer. So, stick with me till the end. I promise the history of a clinical trials is more interesting than you think. So, let's dive in. Here you can see the diagrammatical picture, overall historical events in a clinical research. So we'll see one by one in detail about it. Okay. So let's go back to almost 1,000 years to Avicenna. His name is also called as like Ibn Sina, and he is a famous Persian physician, and he also written the canon of medicine, and it became the most important medicinal textbook for us for centuries. So in this book, the Avicenna defined medicine uh, as like a medicine is the science by which we learn the various states by the body in health and in disease, the means by which health is likely to be lost, and when lost is likely to be restored. Sorry, this shows his scientific approach, studying causes, prevention and treatment systematically. So what this definition is saying, medicine is the science that helps us understanding how the body works in a health and in a disease. So uh, if we lose our health, and how to store it when it's lost. So isn't it like that amazing? Even 1,000 years ago he saw medicine not just as treating illness, but also about prevention and understanding the causes. Now, what's really exciting that Avicenna also gave us the 7 golden rules for testing a new medicines, and believe it or not, these rules look like the way we test drugs in a modern clinical trials. So let's get into all the 7 golden rules. So the first one is the drug must be pure, not affected by heat, cold, storage or a mixing. So the drug should be pure, not spoiled or mixed with other things. So the second one is the test it on a single disease, not multiple conditions at a once. So the drug should be tested on a one disease at a time, not on a people with the many conditions. So the third one is the test it on a opposite conditions to see if it works specifically on the target disease or just relieves the symptoms. So the third one is saying that we need to try on the opposite conditions also, in order to make sure that it really works, not just masking the symptoms and all. Masking means like it is not treating all the symptoms. So the fourth one is the use the right dose, start with the lowest and increase it gradually. So we have to use the right doses, but it has to be start with the small, and we need to increase it gradually. And the fifth one is the observe the time to effect, and immediate versus delayed response. So we need to check the time of effect. Does it really working on immediately, or whether it is taking some time to react? So that we need to observe. And sixth one is the results must be reproducible in a most cases, and it should not be accidental. So we need to make sure that results are repeatable, not just by a chance. So whenever the drug is given to the patients, and it has to show the results in all the patients. so it should not be accidental. And the seventh one is the drugs must be tested on a human body since animals may react differently. So after animal studies, and we have to do the always testing on a humans, because animals and humans don't react in the same way. So overall, that Avicenna says, and this book also says, he influenced the modern trials. So he found the modern clinical trials. He is the founder. So let me tell you all about the first proper clinical trials ever done. It was back in 1747 by a Scottish doctor named James Lind. And at the time, the sailors on a long trips were getting very sick with a disease called the scurvy. So many even died. So the James Lind wanted to test what could actually cure it. So what he has done, so he has chosen the 12 sailors and he divided them into 6 groups. So everybody got the same basic food, but he gave them different extra treatments. That is one group is the, here we can see all the treatment whatever they have he has used. So the first one is the cider. He has given to the first group, and second group got the sulfuric acid, and third group got the vinegar, fourth group got the sea water, and fifth group got the citrus fruit, and sixth group got the spicy paste. Okay, so the results were very clear that the sailors who ate the lemons and oranges got better and quickly, while others didn't show any symptoms. Um, and so this was the first time someone tested treatments in a systematic way, and that's why the James Lind is remembered as the father of a clinical trials. So today even we celebrate the May 20th clinical trials day because of him only. So it is all about the James Lind. So let me take you back to 1937, a medicine called sulfonamide. This is the first antibiotic which is available in 1937. So to make it to a liquid form, the company what they have done is they mixed with the chemical called diethylene glycol. So unfortunately, this chemical is actually poisonous and harmful to the humans body. So it's similar to antifreeze. So when doctors are prescribing this medicine has killed around 105 people, died, and including children. So this was the shocking part. That time the drug companies were not required to test the medicines for the safety before selling them. So this disaster became a turning point in 1938. So the US government passed the Food and Drug and Cosmetic Act, that made it like a mandatory to prove a drug is safe before it can be sold. So like the pharmaceutical company or a sponsor companies, what they have to do, they have to provide the safety data on a IP product. So investigational product information like safety data, efficacy data, and therapeutic use of the data, all the formulations of the data. So everything they have to submit to the FDA for an approval. So this is all about the sulfonamide tragedy. So you guys may get a doubt of this FDA Act. In 1906 they have passed the instructions like uh, adulterated drugs and misbranded drugs are not approved. So that was the act which they have given in 1906, but 1938 they have passed the act is the safety data has to be submitted to the FDA. And the next one is the Nuremberg Code 1947. So after the World War II, the Nazi doctors had done the terrible experiments on a prisoners, so without their consent. So what they have done on the prisoners is like they made them naked, and they have done the trials on seawater in a high temperatures and low temperatures, and even they have done illegal surgeries like bone marrow surgeries and all. So it is a illegal thing. They should take a consent. So without taking consent, they should not, I mean, they should, the subject has to come by volunteer. There should not be any forceful involvement of the subject in a clinical trial. So to prevent such things from ever happening again, in 1947, the Nuremberg Code was created. So it gave us 10 important rules for medical research on humans. So we'll get into the principles one by one. So the first one is a voluntary informed consent of a participant is essential. So the people, as we discussed that in the previous, like in the Nazi doctors, they forced them without their consent, right? So they, what this principle is telling, that the person, I mean people, must volunteer and give informed consent. There should not be any forced into experiments, and the subject, whoever involved in the study, they should have the full knowledge on the trial, whatever the study is going on, what are the risks are there, advantages are there. So everything the subject has to know. And the second one is the research must benefit society, not to be random. So here research should be benefit to society, and it should not like the clinical trial should not test something like useless or a random things. So the third one is the must be based on animal studies and scientific knowledge. So before they are conducting on humans, first they have to do the experiments on the animals, and they should have a scientific knowledge, then only they have to proceed with the human studies. And the fourth one, no experiments if death or a disabling injuries is expected. So they should avoid experiments that could cause death or a serious injuries. If they know, it should be stopped, and they should not continue it. And fifth one is the scientist must stop the trial if it harms the participant. So if the scientist comes to know that if the subject is having if subject is going to face any side effects, then he have the right to I mean the event, like any harmness to the subject, then he have the right to stop the subject, I mean the participant. Subject is subject, participant, and even patient, all these terms are like in a clinical research is the same word. The sixth one is the risk must never exceed than a humanitarian importance of the problem. So here the risk ratio should be lesser than the benefit ratio. So benefit ratio should be greater. It should be greater than the risk ratio, so that it will be useful for the. I mean, it will be useful for the um, to solve the problem. So the seventh one is the provide adequate preparations and facilities for a safety. Um, so researchers, like they should have a well prepared, I mean the well prepared and facilities, like well equipment, and staff should be well qualified, staff should be there. And even the study related subject population has to be there, so that it will be easy to conduct in a ethical manner. And the eighth one is the only qualified scientist may conduct research. So only qualified scientists should conduct the research, and not just anyone. So they should be well qualified and well experienced in this research field. So ninth one is the subject have the right to withdraw any time. So the participant should always have the right to withdraw at any time, even in the middle of the study also. And the 10th one is investigators must stop the study if it becomes unsafe. So investigator must stop the experiments if it becomes unsafe to the subject. And I mean, if it is causing, I mean, if they are, if it is giving the more side effects, then they should stop the study. So this is all about the Nuremberg Code. Now we'll see the thalidomide tragedy. So in the 1950, in the year of 1950s, so a new drug called thalidomide was introduced as a safe medicine for a sleep problems and a morning sickness in a pregnant woman. Actually, this drug is a, sedative drug, not for morning sickness. So, but doctor prescribed for the morning sickness in a pregnant woman. So during the first trimester, the pregnant women will have nausea feeling and vomitings will be there. So that symptoms are called morning sickness. So after taking this drug, what happened within a 3 months, the first trimester is include 3 months, right? So that time there are many miscarriages incidents happen. So those who survived, uh, we can see in the picture birth defects. So congenital abnormalities were found. So those are like limb shortening, heart and ear, eye problems, and even we can see the cleft lip, cleft palate in the images, right? So around 10,000 babies were born with the serious birth defects. So in the year 1961, it got banned, and but US avoided this tragedy due to their FDA reviewer, Dr. Francis Kelsey, who delayed for approval. So after this tragedy, there is rule came like they have to submit all the data after approval only. There will be a post marketing surveillance will be done. I mean, the drug will come to the market once it approved, otherwise it will not come. Again they have to conduct the clinical trial. The drug will go to the, again the same phases, again clinical trials will be they have to do. So this is all about the thalidomide tragedy. So before we get into COVID-19 era, after that thalidomide tragedy, there are two topics which we need to cover. That's Declaration of Helsinki and development of ICH. So those two topics I'll cover in next vlog. So now we'll see COVID-19 era. So when COVID-19 hit in the 2020s, everything is changed. Like lockdowns and hospital risk made like very difficulty to continue clinical trials in a traditional way. So traditional way means like the participant used to come for the research centers for the face to face for the monitoring. So in a person, right? So after that, after pandemic impact, um, so in order to protect the participants while keeping studies running and all, so the Food and Drug Administration and European Medicine Agency, they have quickly issued the emergency guidance that central like a remote monitoring in a clinical trials, like a digitalization. So the technical tools they have introduced, those are the telemedicine platforms, e-consent tools, remote monitoring tools, variables and all. So in order to protect the patients. So this way the real tuning point that pushed the industry towards decentralized and remote clinical trials. So what was only an idea before, COVID, before COVID, but suddenly became a necessity. So this is all about the COVID-19 era. So thank you once again, guys. So we have seen the history of a clinical trials. It shows us like how far we have come and learning from both success and mistakes. And we'll meet in the next vlog. And don't forget to subscribe. And if you have any queries, just ping in a comment section so that I'll reply in a comment section. Either I'll discuss in a next vlog. Thank you. Thank you so much.